Good morning, good afternoon, good evening, and welcome to the second of our webinars highlighting the plant cell focus issue on abiotic stress, plant responses to abiotic stress. Climate change. Okay, we'll let Nan take care of that one. Today we have 3 speakers, and we will be discussing this focus issue on climate change and plant abiotic stress. It came out in January 2023. The editors of this focus issue are listed, and today we have Nancy Eckardt, who will tell us a bit more about this focus issue in just a moment. You can find this issue online at our website. We have 3 speakers today, and our moderator, Kutubuddin, will be introducing them one at a time. And one more note, which is in a few days, we'll post a recording of this to our YouTube channel, and you can find lots of other videos on our Plante YouTube channel, great science content. We want to make a special thank you to the members of the American Society of Plant Biologists who support this webinar series. If you're interested in joining, you can use the code PRESENTS10 to get 10% off your membership dues. We have a question and answer box where you can type your questions, and Kutubuddin will be reading off questions for each of the speakers after their presentation. If you're having trouble, you can email me or try logging out and logging in again. And now I'm going to turn this over to Nan, who will give us a little background on the focus issue. So thank you.
So welcome, everyone. Today's webinar is highlighting authors from the current plant cell focus issue on climate change and plant abiotic stress. And I will again give a brief introduction to this focus issue for our attendees and viewers who missed our first webinar based on this issue. So we had in mind to run a focus issue on plant abiotic stress and decided to emphasize the relation to climate change for several reasons. First, it's widely acknowledged that climate change will have devastating consequences for millions of people. And some of the most obvious effects are abiotic stresses, such as heat stress and drought, with serious consequences for many crops, as well as natural ecosystems and humans. And the latest Intergovernmental Panel report suggested that we need to limit global warming to 1.5 degrees above pre-industrial levels to avoid the most extreme effects. And we probably have less than 10 years in which to accomplish this goal, or in fact, some think that it may already be too late and we need to focus on limiting to a less than a 2-degree increase. So time is running out on the climate change clock. And finally, a couple of years ago now, I heard Joanne Corey at the Salk Institute give a talk on this issue, and she encouraged the audience of plant scientists to spend at least 5 minutes per day thinking about how your research might contribute to mitigating the effects of climate change. And so with that in mind, we thought that the editors of this issue thought it important to add our voice and the journal's voice to those calling for action, and in this small way to provide information and examples of plant research in this area to encourage others to get involved. So this issue includes a commentary from 2 of our editors, Tom Juenger and Paul Verslues, that includes discussion and guidelines for conducting plant drought stress experiments. And then we have 2 vignette-style reviews, one that I organized with over 24 co-authors, where we discuss examples of research aimed at engineering efficient carbon capturing and carbon sequestering plants and developing crops with enhanced climate resilience. And we'll hear a talk from one of those co-authors, Damaris Odeny, today. And another review authored by Paul Verslues and co-authors that discusses unresolved questions in plant abiotic stress biology posed by 15 different research groups. Then we have another 6 in-depth review articles, a couple focused on different aspects of gene by environment interactions. And we'll hear from 2 of those authors today, Jesse Lasky and Andrew Bowerman and Barry Pogson. So they focus on aspects of gene by environment interactions, a couple more on genetic manipulation and breeding for improved climate resilience, and one on abiotic stress memory in plants, and one on the cell biology of primary cell walls during salt stress. And finally, we have 6 research articles, a gene discovery pipeline for complex quantitative traits in maize that uses multiplex CRISPR-Cas9 genome editing, a couple of articles that deal with regulation of stomatal closure, a couple related to salt stress, and one on a vacuolar nitrate transporter that affects nitrogen use efficiency. And with that, I will turn it over to our moderator, Kutubuddin, who will introduce our first speaker. Let's see here.
Okay. Thank you, Nan, for giving me a chance to moderate this exciting webinar. Good morning, good afternoon, good evening to the participants from different parts of the world. So my name is Kutubuddin Molla, and I'm a scientist at the National Rice Research Institute, India, and I'm also working as an assistant features editor for the Plant Cell Journal. I'm the moderator of this Plant Cell webinar on crop breeding for climate resilience, and it's my real pleasure to introduce our first speaker, Dr. Jesse Lasky. So Jesse Lasky received his AB in biology from Kenyon College, Ohio. He received his PhD in ecology, evolution, and behavior from the University of Texas at Austin. He was a postdoctoral fellow at Columbia University from 2012 to 2015. He has been a faculty member in biology at Pennsylvania State University since 2015. He is currently serving as an associate professor of biology at Penn State. Dr. Lasky was the winner of Jasper Loftus Hill's Young Investigator Award from the American Society of Naturalists in 2015. Jesse was also a Fulbright Research Fellow in Panama. And currently, I believe Lasky is spending his time as a visitor in Italy in one of the universities. And at Penn State, Jesse's laboratory studies evolutionary genetics, physiology, and ecology to understand mechanisms of local adaptation to environment and also host symbiont co-evolution. Jesse's talk will be on genotype environment associations to reveal environmental adaptation. And like me, I believe all of you are eager to listen to him. And without taking further time, I would like to invite Jesse for his talk. The platform is yours, Jesse.
Thank you for that kind introduction. Let me get this here. Okay, so I want to acknowledge my co-authors on the review that were part of the special issue. Those are Emily Josephs and Jeffrey Morris from Michigan State and Colorado State Universities, respectively. So this is obviously a session on environmental adaptation and climate change, and we are thinking about this problem of how we could make genetic improvements to plants for future environments, either specific stressful conditions or perhaps more unpredictable and variable conditions. And so one of the approaches and sources of variation that people have relied on for long periods of time are local adaptations that come from populations adapted to different environments. And we have in many widespread crops, we have diverse land races that have spread and evolved over often thousands of years in a really wide range of environments. So these could be important sources of alleles and traits for specific environments. Now, the sort of more new innovation in taking advantage of this local adaptation that already exists, we think, are the use of genotype environment associations. The idea is that these are correlations between allele frequency and environment that allow us to either generate or perhaps test hypotheses about whether a given variant is adaptive in a specific environment. So in this little cartoon I have here at the left, this is a very simple setup of a genotype environment association where we have two environments and we have individuals that are distributed across these environments. And there is this black mutation that confers a fitness advantage in environment A, but not in environment B, and it would be disadvantageous in environment B. And as a result, we have this strong allele frequency difference between environments. So if we test the association or the correlation between allelic state at that locus and the environment, we'll see that the black mutation is associated with the blue environment, suggesting it's involved in local adaptation to that environment. Now you can take these associations, these genotype environment associations, and implement them across the genome like you would for a genome-wide association study for a trait. And in that case, we'll call that an environmental GWAS, where you may be looking all across the genome trying to figure out what locus is contributing to local adaptation or loci. Now, I think a big part of the promise of GEAs, and one of the reasons why they've been so widely deployed in recent years, is that the information required are becoming very easy to generate or obtain. Only genotype information and environment information are required. So it's, of course, becoming cheaper to characterize genotypes over time, and many environmental parameters that we think might be important, especially large-scale climate variation patterns, can be characterized from publicly available databases that you can very quickly obtain downloading from the internet. So you can quickly get these information and test these associations. I think another one of the promises of GEAs is that there's this potential to link causal elements on the genetic side and the environmental side. The idea is that if you were to hypothesize perhaps there are multiple environmental gradients that are important for local adaptation, you could assess the evidence that a given variant is involved in adaptation to a given environmental gradient based on the strength of the correlation in allele frequency. In this case here, let's say you have two different environmental gradients, A and B, the variation from A to B, which is the Cutler, and then the variation from X to Y, which is this either lack of shading or the shading in environment Y. And then you could ask, what's the change in allele frequency across all these environments? And if we compare, let's say, that temperature gradient, if we imagine that A to B is a temperature gradient, we see a strong association with that black mutation and the blue environment. But there is no real change in the frequency of that blue variant if we compare the open or versus the shaded environments. So that's another example, perhaps, of how the GEAs may be useful, is tying a variant to a specific environment that we think might be important for selecting on it. Now, there's a few, I think, often overlooked assumptions of genotype environment associations that we wanted to cover in our paper. And the idea is that a lot of people have been using these associations, but perhaps often there's not an explicit consideration of some of the favored hypotheses or perhaps assumptions underlying them. So I'm going to run through a few of these. The idea is that the favored hypothesis is one that is a situation, if it's true, then the situation is favorable for the use of GEAs. But if the unfavored hypothesis is true, it's not as good to use GEA. So in the first case, the favored hypothesis is just that your individuals that you're studying inhabit different environments. It's very simple. And if your individuals do not inhabit the same different environments, if there's very little variation in environment, then of course that's not a good system for studying environmental adaptation. But let's assume they do. Let's assume that favored hypothesis is true. We'll move on to the next favored hypothesis, which is that the populations are locally adapted. That is, that genotypes have more fitness in their home environment than foreign genotypes. So if we look in that blue environment and we say fitness scales with the size of the circle, the gray genotype, which is more common there, has higher fitness in that environment, and vice versa with the white genotype in the red environment. Now, this is important because populations aren't always locally adapted, and they're not always locally adapted along an environmental gradient that a researcher might think is important. So if the populations aren't locally adapted at all, this may not be a good system for studying local adaptation. But let's assume that they are locally adapted. To fulfill this promise of the GEAs, perhaps tying together causal genotypes with causal environmental variables, it requires some degree of independent variation between environmental dimensions. That is, the favored hypothesis is that they are not confounded with each other. That is, the multiple environmental gradients. If there is confounding, and there often is some degree of confounding or covariation between environmental variables, it becomes harder to separate their influence on allele frequencies. Now you can have some very careful study design to try to decrease those correlations, but this is a challenge for this approach. Again, and so the next favorite hypothesis that I want to mention is that you have oligogenic local adaptation. That is, that there are few loci of large effect that contribute to local adaptation. In this case, the black adaptive mutations are shown, their size is scaled according to effect size. And you see when we have this oligogenic adaptation, there's one locus, polygenic adaptation, many small loci. When we have many small loci, it becomes harder to implement or harder to learn about adaptation from GEAs because you have so much covariation across the genome. Careful study design can reduce that. Also, when you do have that polygenic adaptation, it may be a situation where the GEAs are better suited for genomic prediction of whole genome phenotypes. The next favorite hypothesis is that we don't have confounding with population structure, that is, confounding between the adaptive mutations and neutral ones. Of course, though, we often have limited gene flow. We often have limited gene flow across environments. And so when you have that, you can get a buildup of population structure or differences in allele frequency across environments. And so that long-range linkage disequilibrium that builds up makes it harder to separate variation across the genome. This is a common issue with GWAS for phenotypes as well. Again, study design can help reduce this, but there's a degree to which you're limited. And then the last one I want to mention is that we have genetic homogeneity. That is, let's say, a single mutation at your locus that is causing the adaptation, as opposed to independent mutations or independent mutations at different loci causing a similar phenotype. So when you have the heterogeneity, it becomes harder to learn from the GEA because each variant is rarer. And so these observational correlations like GEA or GWAS in general have less power for rarer alleles. Now, I just want to mention, we wanted to mention a few areas of recent progress, or sort of the directions which we saw the field headed with GEAs. And I'm going to sort of classify things into forward ecological genetics as one category. That is where we're scanning the genome and trying and looking at variation and trying to find variation that might be adaptive. That is the environmental GWAS approach. Now the environmental GWAS has been around for a while, basically since the high throughput sequencing came on. And here's one of my earlier studies that's an example of this. But in more recent years, the field is moving to the direction, I think, of trying to ask whether environmental GWAS peaks also co-localize with phenotypic variation to try to increase our confidence that there is actually a phenotype associated with the environmental GWAS variation. So this is an example in Populus from Evans et al., where they showed that this phenology peak from a phenology GWAS on the top co-localizes with a genome-wide peak for environmental variation. That is, this variation that underlies or is strongly associated with phenology also is strongly associated with the climate of origin of these populous genotypes. An alternative approach that we're going to call reverse ecological genetics starts with known variants that have some known phenotype, and then asks whether there's evidence using the GEA, whether there's evidence that local adaptation is explaining this variation that already exists. So this is a nice example from Arteaga et al. That was published in Plant Cell a couple of years ago. The phenotype here that they're interested in that varies in Arabidopsis in Spain specifically is this variation in trichomes on fruits and pedicels. So they show with these introgression lines that these loci that are introgressed are causal for these traits. And they then ask, is variation in sequence at these loci associated with environment, which might suggest that that environmental variation selects for alternate alleles and alternate traits at these loci. And what we see here on this map is that the red regions, which are drier and usually lower elevation, are associated with the presence of the trichome alleles. Those are the ones in red or orange. Those are either the circles or the triangles. So that suggests that perhaps some selection associated with rainfall in the spring when these are flowering, could be selecting for trichomes in the drier sites. Another recent area of progress that we wanted to mention briefly was genomic prediction. And this is an example of our work from some years ago now, where we looked at sorghum land races. We found SNPs that were associated with growing season length, and we then used that to predict relative drought sensitivity among genotypes, and saw that it did predict sensitivity to drought, in this case here on the right, measured according to change in the panicle head weight. Okay, so there are a lot of technical considerations for GEAs that we didn't want to get into here, but we did want to point to a number of recent studies, either simulation-based that compare methods or reviews of these methods. And so there are important considerations like how to deal with population structure, how to model environment and genotype. Now, so one of the things we wanted to mention is that we feel like the current status of the field is that a lot of studies have been focused on either of those sides of the coin of forward or reverse ecological, forward ecological or reverse molecular genetics. That is, either generating hypotheses from GWAS, but often not testing them enough in isogenic backgrounds, or generating hypotheses that are based on known effects of mutations that are studied in molecular genetics, but with a lack of testing natural variants in ecologically relevant conditions. So we're suggesting that we try to integrate these approaches, combining the hypotheses that are generated from environmental GWAS with testing of these in isogenic backgrounds, and also being able to perhaps use the molecular genetics information to prioritize genes that come from the GWAS, as well as taking important known clone variants, natural variants, and testing them in ecologically relevant conditions. So I just want to briefly mention some work that we had done in just a few seconds here. So we have looked at a natural mutation identified by the Ageta lab at Purdue that causes Striga resistance. It was identified from biparental cross and in a few lines. We then went and former postdoc of mine, Emily Bellis, studied variation at this locus and found in the land races, the larger set of land races, and found that this deletion allele or deletion alleles that are identified by the Ageta lab actually are more common in Striga-prone regions, suggesting that's the GEA, suggesting that there is a local adaptation there. But that there is this limitation in frequency of those, even in the Striga-prone regions you see on the right there. Even in the most Striga-prone regions, the allele frequency of the resistant mutations do not increase the fixation. They're still fairly low, suggesting perhaps trade-offs associated with that. And that generates this ecological hypothesis that there are maybe trade-offs. And so I don't have time to talk about those. We did a genome scan for this environmental GWAS. And I'll then just get to my conclusion, which is that we still need more extensive empirical validation of environmental GWAS, those hypotheses that are generated. And we think that in the future we can do better by integrating ecological and molecular genetics to get the information out of the GEAs to learn about adaptation. Thank you for listening and attending.
Oh, thank you very much, Jesse, for the great talk. And I guess it will be a great resource for those who want to study and learn about the molecular basis of local adaptation and testing hypotheses. And also thank you for being on time. So it's now time for taking some Q&A. So there is one question, and for the participants, if you have any question, you could write in the Q&A box, and I would read it loud to the speaker. So the first question is from Ali Abdel Kader from Algeria, and he asked that what is better, studying the interaction genotype environment or specific gene with the environment? For example, quantitative trait loci with environment.
Yeah, so it's a persistent sort of diversity there in approaches and strategies that I think maybe is being captured by the questioner. And I think that it really depends on the specific goals and the specific context and questions that one has, whether or not one needs to understand how variation in an individual gene causes change in fitness across environments, versus maybe whether one is more interested in understanding the ecophysiology that leads to changes in performance across environments of different genotypes. So I think it's really sort of context dependent, but they're sort of all, both important general areas of inquiry.
Okay, so I don't see any other question now. So probably some question will pop up later. Yes, there is one question from Rahil. Which sorts of population is best suited for GWAS studies in maize? Example, inbred lines or Nils?
So I guess so when we're, in terms of nils, I guess, I haven't been involved in studies where we're doing GWAS with a very big, diverse library of nils. It would be a useful resource because it would not have the long-range disequilibrium, but I'm not aware of that. Maybe we're thinking about RILs as being the population of interest, and I think it's context dependent. There's benefits from the GWAS, large amounts of diversity captured, perhaps greater resolution, but the long-range confounding with population structure, long-range LD across the genome is still a problem that is broken up by experimental recombination when you generate RILs.
Okay, great. Thank you. So there is another question, and this is the last question I'm taking because we have to move forward to the next speaker. So this question from Janely Livesay, with respect to environmental GWAS, how could that be used to translate to disease pressure?
So in our case, we were looking at Striga, which is a parasitic plant, but you could think of it like a disease. And what we had to do was come up with an estimate of the prevalence of that parasite, or you could think of the prevalence of that disease across the continent of Africa, and then we linked that to genotype. So maybe different diseases or different parasites or herbivores, let's say, are different pests. You maybe would do that in different ways depending on what kind of information you had on the distribution of that. But the underlying hypothesis that is important to be explicit about is that the populations in regions that are more subject to the disease are adapted via resistance or tolerance. That has to be true to have the environmental association work, because it only works if there is some local adaptation.
Yeah. Okay, thank you. Jesse, so I would request you to look at the Q&A box, and there are some questions, so you could just reply them in the box. Okay, thank you. So it's time to listen to our second speaker of this webinar. And before I hand over, it is my real pleasure to introduce Dr. Damaris Odeny. Damaris earned her PhD in plant genetics from the University of Bonn, Germany, and completed postdoctoral training from the Max Planck Institute of Plant Breeding Research in Cologne, Germany. She is a plant molecular breeder with research experience in Africa, Asia, Europe, and USA, and also has led and implemented genomics projects in cereals and legumes. Damaris led the development of genomic resources in several orphan crops. She works closely with other disciplines from national, private, and international institutes to develop cutting-edge genomic resources for all ICRISAT mandate crops like sorghum, pearl millet, finger millet, groundnut, chickpea, and pigeon pea. Damaris is also passionate about mentoring young upcoming scientists and has successfully mentored more than 50 scientists in her career to date. Damaris Odeny has also been an award fellow in 2010, and she is a strong face of women in science and leadership in Africa. So and now currently Damaris is actually working as a scientist in ICRISAT, Kenya, Nairobi. So without further ado, I invite Damaris for her presentation on genomic resources for orphan crops and enhancing climate resilience through crop wild relatives. So Damaris, over to you.
Thank you. Thank you so very much, Kutub, for that introduction. And I also just want to thank everyone, including Nancy, for providing me this opportunity to present my work here. I'll go right into it. I want to start my presentation by drawing two parallels in terms of the reality of underutilized resources. And the top one there I'm giving is, a young lady called Miriam, who is now 15 years old, has never been to school. And finally, she learns about school and education, and she wants to be a lawyer. And so at 15, she needs to go through basic education, high school, university, go to law school. That would take her at least 15 years to become a lawyer. But she has to compete with much younger and perhaps more resourced students who are more privileged than she is. And that's similar to what underutilized crop species are going through. And I'm just giving an example here with the finger millet, which is one of the crops that I work with, which is an underutilized crop, quite a precious crop, but has not been, does not have much genomic resources. But suddenly it has been found out that it's climate resilient, it's highly nutritious. And so breeders are looking to release varieties from it. So there's just that wide gap that requires a lot of things. And I've just highlighted a few things, a few components there that needs to be done. You know, you need to understand the basic genetics, its adaptation, go into advanced genetics, and then even try to understand the physiology, agronomy, the consumer preferences before you even get to release a variety that would be impactful. So that's the story of what we've gone through with finger millet, and I'll just say a little bit about finger millet. It's actually a very nutritious crop. It's native to the African continent. Must have been the earliest domesticated cereal, estimated at about 5,000 years ago. And it was domesticated from its wild progenitor, which is also a tetraploid. So finger millet is a tetraploid. But it's quite resilient to climate. It's drought tolerant. It thrives under low input. It also resists quite a number of biotic stresses. And it's really high in micronutrients. It has decent amount of protein, fiber, fatty acids. It also has superior malting quality. Most of these qualities have not really been exploited, and it's also gluten-free. So you can imagine with the current changing lifestyle where people are looking to eat healthy foods, finger millet is a really important crop to go for. I'm just comparing, I'm putting on that screen there, comparing finger millet with other most utilized crops like wheat, rice, maize. And you can see it has almost 10 times more calcium than some of those crops. Magnesium, it's having quite a high amount if you compare with maize, and potassium also quite significantly high. And just to mention that, you know, this is just from random analysis of some genotypes. You know, if we go into looking at variation across different genotypes, you can find even higher amounts. But the challenge is it's a tetraploid, so you can't rush the development of genomic resources. The AA genome donor is known, but the BB genome donor is not known. So still some work needs to be done there. And the production challenges are a parasitic weed, which luckily for me, Jesse already mentioned, Striga hamontica, which is a very a major stress in most of African locations. But in this context, I'll talk about Eastern and Southern Africa. Blast disease is also a big problem. And of course, you know, even though we talk about drought resilience, with the current changing climate, there are times that you do not get rain at all. So sometimes you still need to look for even more drought tolerance. And also there's always a balance. You give up when you start to increase yield, then the crop starts to reduce its adaptation to drought. So drought is a standard trait that we look for when we're doing crop improvement in finger millet, drought tolerance. So where we started with this crop a few years ago, I would say now about 9 years ago, is start doing whole genome sequencing and annotation, just to be able to ensure that we have a robust set of molecular markers of genomic resources, that we know what genes are. We are able to figure out where some traits are associated. So we estimated the genome size. We we also annotated the genome just using the basic resources that are available. And at the time, we just used Illumina sequencing, but we've been able to improve the resources with time. But beyond just doing the whole genome sequencing, we also used the resources that were generated from the whole genome sequencing that actually does allow us to be able to now use molecular markers more routinely because we have a reference genome. But we went further because most of the breeders who are utilizing this crop do not have access to very advanced computing facilities to be able to do things like genotyping by sequencing. So we were able to also develop quality control markers where we developed a set of 20 very highly informative markers that are able to distinguish the different genotypes at very high informative rate. And I'm just showing there in this figure the performance of some of the QC markers that were generated for it. And just recently, we were also able to develop mid-density panel markers that were initially about 2,500, but of course, with validation, we lost some. And so now we have about 1,700 mid-density panel markers. Now, what those resources have helped us do is we are able to genotype vast numbers of genotypes, and we are able to do other activities like association studies and looking at all these traits of interest, like micronutrient content, looking at iron and zinc. But for this presentation, I will limit it to how we've been able to use these genomic resources to utilize crop wild relatives to look for traits of interest. So the focus traits are striga, blast disease, and drought, as I already mentioned. So we were fortunate in 2015 to be able to receive funding from Crop Trust, and we've been fortunate since then to continue to receive funding from different sources to be able to address these three major traits that are a problem in finger millet. And as I was mentioning, finger millet is trying to be at the same stage with all these other quite advanced cereals that have most of the resources. So we are really trying to fast track and also use comparative genomics sometimes to be able to fast track and identify the traits of interest. So we learned from sorghum, which already had identified novel sources of resistance for striga from crop wild relatives. So we went ahead and looked into the collections that we had available from the Kenyan Gene Bank and did some screening. And what I'm showing on the screen here, you can see, this is just for a few genotypes, but you can see that the ones highlighted there are wild relatives of finger millet that were showing, they were supporting much less striga on their roots when they were infected with striga. So some of these were probably not as stable, but this particular one, less 10, was quite stable, showing resistance to Striga hermonthia. We also went ahead and did histological analysis, and we could observe something that looks almost like mechanical resistance, and mechanical resistance actually has also been reported in the other cereals that have been well studied, such as sorghum. And so this was quite promising. So the less 10, which is showing on your screen as the wild resistant there, seems to have a really good resistance to striga, but also we were able to identify Okhali-1, which is already known. It was almost used as a control that shows some level of tolerance in the field. Then we went ahead and were able to use to generate biparental populations, and now we are doing biparental mapping alongside genome-wide association studies, also using larger panels. Now, I'll also quickly go through another trait which we've used, crop wild relatives, and that's blast resistance. So blast disease is also one of the major diseases in finger millet, and it's the same blast that causes blast disease in rice. Now, when we screened a number of crop wild relatives, we were able to identify most of the ones that were resistant were actually wild. If you look at that screen, the ones that have W under the most resistant are actually wild relatives. Now, in another study where we had already crossed wild and cultivated, we actually went ahead and we were able to map QTLs in a biparental population that is interspecific, and we were able to map for resistance to blast. So those are just a few examples. One of the things that we are doing is really doing capacity building, because there are very few people who are involved in this research. If we want to have an impact, then we have to be aggressive in capacity building, and we're doing it at different levels. But the summary of what I'm trying to, what I've had the opportunity to say there is that underutilized crop species are currently creating very exciting opportunities, and there's attention to them, and also affordability of genomic data makes it easier to manage linkage drag, which was usually the limitation that we had in the past. But obviously, sometimes there's need to be to balance between just going ahead and generating genomic resources, because most of what you need is actually more precise phenotypic data. And so there needs to be a balance between phenotyping and generating genomic resources. And as I mentioned, we need to do more capacity building and more collaborations in upstream research for us to really understand the biology of the different stresses that we are working with. And to finalize, I'm just saying, thank you to our funders. Thank you to everyone who was listening, and to the collaborators who are involved in this work. And thank you for the opportunity. Yeah, thanks.
Yeah, thank you very much, Damaris, for the excellent talk. So yeah, now it's time for some Q&A. So there are 3 questions in the Q&A box, so I'd read them sequentially. So Karin asked, does finger millet contain compounds that reduce the absorption of micronutrients by human?
Yeah, that's a good question. So there could be, if you compare finger millet and sorghum, for example, sorghum seems to have more antinutritive factors, and finger millet has less. The thing is, we haven't really explored that much to look at the extent and the variation across different genotypes to make a conclusive argument on that fact. But yeah, the comparison between finger millet and sorghum is that finger millet has much less, and the digestibility is actually much better than if you were to compare with sorghum.
Okay, great. So Daniel asked that, are you working with any company to finish the sequence for future studies?
Yeah, so actually the final high sea level assembly of finger millet is out actually, and you can access it on Phytosome. And the publication will also be out in the next few days, because it's been accepted for publication. But that aside, there are other reference genomes for finger millet that are available. They're probably not at chromosome level, but they can be utilized. But the one that is available at the highest level is actually accessible. So you can use it. You can use it for calling SNPs and all that.
Okay. So then Mridul Chakraborty asked, what is the scope for ploidy manipulation, supported genome edition in finger millet?
Yeah, there's huge scope for so many things in finger millet. I think some of the challenges that we are struggling with at the moment is, the protocol for for genetic engineering is not really optimized. But I mean, there's everything to be explored in finger millet right now. There's just so many things that can be done. And if there are resources, most of these aspects can be exploited and tested as much as possible.
Okay, great. So next question is Nicholas. Nicholas asks, can you comment on the importance of traits such as lodging, shattering, and seed size? Those are really related with domestication for finger millet.
Yes. So, I mean, one of the things that we all have to deal with when we're just working with underutilized crop species is that, you know, before you get it into mainstream breeding, you have to deal with all these domestication traits. But at the moment, actually, we think that we've figured out the shattering gene, and hopefully, if we're able to get the genetic engineering working well, I do know that Katrine DeVos has done a lot of work on this, and she might be publishing it soon. But all those domestication traits are still a problem. The seed size is small, but the variation that we see for seed size is enormous. If we look at collections that are available, for example, in South Sudan, you have finger millet landraces that have really bold seeds. So there's scope there to increase the size. But yeah, I mean, things like shattering and lodging are still a problem, but I think the diversity that exists gives us the opportunity to be able to utilize alternative genotypes that have resistance to these traits by introgressing them into the mainstream breeding.
Yeah, thank you, Damaris. So we have to stop here. The great discussion that was going on. Now we have to move to the next talk. This talk will be delivered by Andrew Bowerman and Barry Parkinson on an interesting topic entitled Abiotic Stress Targets for Modern Genetic Manipulations. So before I go to introduce both of them, so I would request Damaris to look at the Q&A box, and there are some questions, so you could answer them there. Thank you. So coming back to introducing the speaker, Dr. Andrew Bowerman, who has had a career working to improve the quality and reliability of yield in wheat. He has done PhD from the CSIRO and the Australian National University with Professor Barry Parkinson. During his PhD, Andrew investigated grain endosperm, starch biochemistry, and examined how starch phosphate ester content altered starch processing qualities and also affected germination and growth. He also worked on gluten polymer quality and grain carotenoid content using both conventional and genetic engineering methods. Andrew joined Professor Barry Parkinson's group as a postdoctoral fellow, and his postdoctoral work has focused on dirt respiration and energy use efficiency of wheat growth in an international wheat yield partnership project. And his work has branched into retrograde signaling for drought tolerance of wheat via the SALON-PAP pathway. And coming to Professor Barry Parkinson, Professor Parkinson is a fellow of the Australian Academy of Science and currently serving as the director of the Australian Research Council Centre for Fruit Crops. The Centre for Fruit Crops is focused on training the next generation in the new genetic technologies for improving crop yield and resilience in response to changing climates, that inclusive of societal expectations. He is also an ARC laureate fellow in synthetic biology for developing smart crops that are higher yielding and more resilient in both good and bad seasons by precisely switching on and off resilience. This work builds off his research, which has revealed pathways and processes that regulate growth, development, photosynthesis, and tolerance to drought, a process known as retrograde signaling. Earlier, he has also served as a senior editor of the Plant Cell Journal. He has also been conferred with ASPB Top Cited Author Award for the Plant Cell. And I also want to mention here that Professor Parkinson's group has received 4 national awards for research excellence, 2 awards for teaching and supervision excellence, and 6 students have received university medals and awards. So without taking further time, I would like to invite Professor Parkinson and Andrew for their talk. So over to you, Mary, could you please play the video?
Hi everybody, and thank you for the opportunity to speak about how genetic technologies can be applied to the challenge of climate change. The idea that climate change is simply hotter and drier masks the reality that the major change in climate the world will see is a rise in local climate variability. The likelihood of extremes is increasing on both sides of the temperature and rainfall distributions, with records for heat, cold, drought and rain rapidly being broken. At current emission rates, we've only 9 years left until we've reached the IPCC goal of limiting global warming to 1.5 degrees Celsius, with CO2 emissions returning to pre-COVID levels. Ice loss in Antarctica and Greenland has been underestimated. The global water cycle is accelerating, and 2022 was the warmest year on record in 28 countries. We rapidly need to make changes to both adapt to and mitigate the effects of climate change. But adaptation effectiveness decreases as the climate changes, and only 8% of global climate finance has been directed at adaptation mechanisms. The challenge this presents to food security is clear. Estimates of loss from single stress types in future are disastrous. Up to 13% reductions of yield with major crops due to a temperature rise of 2 degrees Celsius has been suggested, while the changing frost window has seen late-stage losses of productivity, as exemplified by the April 2021 frost events in Europe, where 80% of France's fruit trees, oilseed production, potatoes, sugar beets and vineyards were damaged. Changes in rainfall will see major losses in the US and Canada in coming years in wheat and soybean production, while Southeast Asia may see a rice yield reduction of up to 20% by the end of the century due to water disruption. Overall, a pattern of longer, harsher droughts interspersed by shorter wet periods with greater risk of extreme rainfall is predicted, the impacts of which Eastern Australia has seen in the last few years. Rainfall pattern changes will see soil degradation and salinity rates increase, along with a greater dependence on groundwater sources. Alone, these individual changes to climate are worrying enough, but the chances of growing seasons with multiple abiotic stresses arising is increasing, including different, sometimes opposite stresses occurring in the same crop. As an example of the changes to production this has caused, wheat cultivation in Mediterranean environments starting earlier due to the potential reduction in early winter rains. This requires deeper sowing to utilise deeper soil moisture reserves, but in turn this can delay germination and then extends the flowering period later in the season. With the change in likelihood of extreme events, this increases the risk of both heat and frost stress occurring during flowering. Rather than focusing on a single stress type for improvement in the face of climate change, plant scientists and breeders need to work together to approach this question on two different fronts. Expanding the ability of crops to maintain yield across a wider range of year-on-year variation will provide a more secure base to crop production systems, whilst improving the resilience of crops to manage severe abiotic stress events will reduce the penalties these normally inflict. Together, these approaches can minimise the negative years and enhance the good seasons. Improving yield stability must be a priority, targeting the establishment and early vigour of plants, improvement and stabilisation of resource acquisition, and protection of energy-related processes. Examples are improving the ability to sow deeper for improved access to moisture by using new GA-sensitive RHT dwarfing genes. Such a change in wheat is predicted to increase yields in future years by up to 20% in Australian conditions, due to the combination of improved germination and early vigour. In turn, this enhances water use efficiency by increasing ground cover and improves light interception. Improving root structures and hydraulic conductance will generate materials better capable of taking advantage of water and resources. Enhancing retrograde seedling pathways from chloroplast and mitochondria to the nucleus to better protect photosynthetic machinery is a high priority, as is enhancing respiration efficiencies and stabilisation of rubisco-activase at high temperatures. Concurrently, improving resilience to abiotic stresses will be essential to ensure plants survive in a fit state to generate a final yield. We should seek to modify the sensitivity to and timing of ABA's effects on stomatal opening and transpiration, which may provide protection in the event of drought stresses, especially by restricting such changes to specific tissues to to prevent any general yield reduction. Alteration of specific aquaporins to optimise hydraulic conductance under different drought and water stresses. Engineer cell membranes to resist heat stress and modify production of ROS scavenging enzymes. And finally, modify salt exclusion in a tissue-specific manner using KT genes to maximise capacity to succeed under salt stress-inducing conditions. It's important to note that as more and more of these targets are optimised, of especially stress-responsive components, these pathways are often involved in several stress response mechanisms. Evaluation of the potential for changes to antagonise other areas of importance cannot be ignored. For example, salt exclusion mechanisms may cause issues for resource acquisition and for drought or water stress mechanisms. Other systems, such as leaf establishment, may not be affected, while there may be some aspects that are enhanced by these changes. Similarly, we can theorise about the antagonistic, neutral and synergistic relationships between drought and water stress responses with other components, where there may be enhancements in growth and resource acquisition. Some modifications could be entirely advantageous to other systems, such as establishment of bigger changes. In order to meet these needs to improve crop species, normal background allelic variation will not be sufficient. We'll need to import new alleles from different species, improve the pace at which we can genotype with methods such as hyperspectral prediction models, and further rely upon genomic selection and traditional breeding platforms. New technologies will need to be deployed using both GM and gene editing capacities. We'll need to use stacked transgenes for synthetic stress detection and response pathways, and implement the use of homologous recombination methods to permit repeated, single locations that will simplify crossing to elite varieties and interbreeding panels. We can target stress-responsive elements either systemically or in specific tissues, cellular locations or time points, such as for the HKT salt exclusion changes. Species-agnostic transformation technologies such as Wushel baby boom and nanoparticles for delivery will become important. And use of spray-on encapsulated double-stranded RNA to modify expression of genes in leaves as and when appropriate for a given season, such as to modify ABA receptors prior to drought stress arising in plant. We could generate smart plants that use the accumulated abiotic stress signals to determine likelihood of primary crop success, and then modify metabolism and nutrient mobilization to maximize secondary production, such as fodder or hay, when such primary crop success looks poor. We can optimize gene editing for delivery, such as producing minimal-sized CRISPR systems to better fit into nanoparticles for transformation of pollen or the meristem. And we'll need to streamline methods to improve the pace at which we can generate these new genetic materials. However, there has been a major issue in transitioning such changes from the lab to the field. Some estimates are as low as 2% of glasshouse phenotypes translate to field conditions. Researchers need to work with breeding companies to allow the introduction of engineered changes into the breeding platforms that have to date generated the major advances in yield potential. This will allow these novel materials to be tested and evaluated in many different conditions and genetic backgrounds, rather than the current methods that often don't use either elite materials or use only single environments. Only by incorporation of modern breeding methods with novel genetic variation can a full picture be developed of the value of such engineering programs. Acceptance of these technologies and advances by the public, and especially farmers that will actually grow these crops, is not guaranteed. If farmers are not willing to grow the crops, or consumers are not willing to buy and eat products made from them, even the most scientifically compelling technologies will be unlikely to proceed to market. Overall, evidence suggests the public's concerns are in multiple areas. Farmers' rights and consolidation of multinational biotech firms' control of crops is an issue for the public outside the normal concerns of GM and biotechnologies themselves. While many consumers' concerns don't map directly into a distinction between GM and the non-GM. Instead, focus on minimal processing, local, nutritious and additive-free food production is prevalent. Gene editing is seen as less concerning than genetic modification. There is evidence that there's more willingness to accept novel crops generated for resilience if there's a benefit to the farmer, especially smallholders and family enterprises, and if there are other advantages such as in a reduced environmental impact. We recommend that scientific experts and science communicators should find ways to engage in genuine and meaningful dialogue with various publics, including those working in agriculture and community members at the earliest stages of research and on an ongoing basis. We should promote the consideration of risk and benefit of the specific crop in question, how the product fits within current or alternative ag systems, and the contribution to food security and environment of the crop itself. Transparency is critical. Technical details about the science and general economy from such advances are not. The generation of a social license to operate that includes a community-inclusive underlying structure will hopefully assist improve the regulatory systems globally to develop crops from the advances we've suggested here and advance the cause of food security in the face of the climate change we're already witnessing. This presentation is an overview of the article we published in the abiotic stress edition of the Plant Cell, and we thank the Plant Cell for this opportunity to highlight our review. I'd also like to thank my funder, the ARC Training Centre for Future Crops Development, which hopes to train the next generation of researchers to address these challenges I have highlighted. Contact the centre if you'd like to join or collaborate with us.
Okay, thank you very much, Andrew and Parkinson, for this a nice talk. So it's you have some question in the chat box. So someone asked that what are the plant abiotic stress challenges from the changing environment? So,
that's a very broad and open question there. I mean, as the climate change is getting worse, as these changes are developing, we're getting more longer drought or high rainfall events. We're getting higher temperatures. We're getting lots of frost events. But I think the biggest challenge for abiotic stress events in a single season is the fact that with the expansion of the number of events that are occurring, the chances of getting different stresses occurring in the same crop that can confuse the systems that are there to protect crops is becoming a real problem. So having, for example, when we're talking about problems that are causing delays in flowering time, what we're getting with later frost events is that now around the flowering time, we're getting both heat stress at flowering, and we're also getting the chance of frosting occurring at flowering. How a plant prepares for what it thinks is going to be a very hot period, but then has to immediately swap over to the opposite and deal with a frost event at flowering time. That's a huge question, and I don't think we've got a good answer on that yet. But so from that perspective, I think the biggest challenge is how we can make crops smarter, or how we can help the crop to make those decisions, because we can see these, you know, if we get a bad forecast coming, we've got to have the opportunity and the capacity to help the plant make that decision.
great. Do you have anything to add here? Oh, so you need to unmute yourself. You're muted. Yeah, thanks.
The major challenge we all tend to picture with climate change is it's a hotter, drier climate, but it's not. It's a more complex climate. It's more extreme floods, it's more extreme droughts, and it's this concurrent challenge in the same season of frost events with drought that's going to make it so much harder for the breeders. And I think that's the shift in mindset that we have to have. We're not just looking for a drought tolerance allele. We're not just looking for a frost tolerance allele or salinity. We've got to be able to stack things and breed for things and bring together traits faster than we ever have before.
Yeah, thank you. So let me ask you a question that is open to both of you. So Often, abiotic stress tolerance is considered as a polygenic in nature. So what do you think, how feasible it is to use modern genetic manipulation, like genetic engineering and the genome editing, to engineer our crops?
I think there's a lot of possibilities there, especially the new technologies are making this a lot faster to generate these kinds of things. I think there's lots of things that we can. Yes, there's polygenic responsiveness there that means that you're often working on very different pathways at a time to try and deal with these things. Interesting ideas that show a lot of promise are things like actually modifying expression in the plant on the fly. This idea of being able to spray on double-stranded RNA that's in bound to clay nanoparticles that gets incorporated, and you can turn on and off expression patterns in leaves. Maybe you can see that there's going to be a week of very hot weather coming in the forecast. We might be able to get to the stage where we can actually turn off or turn on structures that are going to protect the plant for just that 2 weeks, and maybe even change it back again using a different spray on. I think that kind of technology is the really interesting stuff that's coming. And I think synthetic biology circuits that we can produce that are going to be able to have smart switches involved is going to be a really interesting thing to look at in the next decade.
I'd also add that as technology is improving, our ability to integrate a lot of wild genes simultaneously is improving with the power of genomic selection. And gene editing is offering us the chance to edit 20 different genes at a time. So I think our ability to deal with complex traits is more based on our ability to deal with the knowledge and the wisdom of what traits to stack together for this future climate complexity than it is the technology. Over the next 5 years anyway, I think the technology will cease to be limiting. We're already seeing papers coming out every other day on gene editing in every other crop. It's now a case of what are the smart targets that actually give a yield increase, not just a more drought tolerant plant.
Thank you. So there is a question from Jeffin Kosha. So he asked, in North India, we faced heat stress last year, and things look similar this year too, which have huge impact on wheat yield. From breeding point of view, what would be the short-term goals a research community should focus on to breed terminal heat tolerance in wheat? Because you have long experience working in wheat. Yeah.
That's a very good question, and it's probably a question that we're trying to answer in Australia a lot too. The terminal heat waves tend to bash our crops around a lot as well. I think one of the most basic things that we need, that maybe we can engineer a bit more, is flowering time. That it's always the thing that comes up in every phenotypic screen, or GWAS. Flowering time's always the thing that pops up. It's unfortunate, but at the same time, it is an answer. Maybe there's a way that if we can see heat, major heat stresses coming, that we can either delay flowering time or accelerate flowering time with enough notice. That, of course, is dependent on really good meteorological information coming as well. In terms of other heat defense mechanisms, modifications to the trying to change the lipid content or the lipid types in the cell wall can produce some major changes in heat tolerance. But yeah, Barry, any other suggestions?
No, I think you've covered it.
But
Okay,
in terms of when it comes to short-term goals, these are, if it's short-term your goal, then you're going to need GM, and you're not going to be able to do it any other way.
yeah, I agree. So, there is an interesting question. So this would be the last question, although there are other questions in Q&A, but we are short of time. So Nitish asked that, as you told about dealing yield as well as stress resilience at the same time, What should be the approach to deal and which one should be first targeted, as both are sometimes antagonistic? And what should be approach to deal with drought as well as submergence in rice simultaneously, as we face this now every year? So this is kind of two questions.
Yeah, two questions. And I think they're on exactly the kind of theme that the paper was all about. That combinatorial stress is our biggest problem coming forward. I think when it comes to, as we said, dealing with yield and stress resilience, I think we need to build partnerships. We don't have the time to make a decision to work on one. We have to be building the most important thing we need to do is build partnerships between researchers and breeding companies as each group is developing something in either a stress resilience area or in a yield stability area. We need to be feeding them into the same groups so that we can begin testing them in combinations as soon as these things are ready. Pushing them into that breeding site, that modern breeding system is exactly what we need to do. Yes, there's going to be a lot of things that are going to be antagonistic, but the reality is we don't need synergism. We need neutrality. We just need it not to hurt something else. So whatever things we're working on, we need to be working on them at the same time as somebody else in a partnership working on other areas. When it comes to drought and submergence problems in rice, I know that rice is one of the better crops in terms of submergence tolerance. So as a wheat person, I get rather jealous at times of the submergence tolerance that rice has. I think there are similar pathways in between drought and submergence tolerance at times, in that there's going to be feedback into photosynthesis and respiration problems as well there. But I couldn't give you the best target there. Maybe Barry's got a lot more experience in drought and retrograde signaling stress responses. Maybe he'll have some things to add there.
So what I would add is that, yes, yield and resilience are often antagonistic, but they're not always. Evolution itself has brought about this complexity of signaling pathways, right? ABA is both a developmental hormone and a stress hormone, to put it simplistically like that, right? And stomata open and close, and open a little bit and close a lot and close a little bit. So it's very rare. It's, as scientists, we treat these things as switches. And for the last 30 years, we've been knocking out mutants and saying that controls drought because it controls stomata. The next generation of crops isn't about the on-off switch. It's about the dial, the rheostat, the thermostat, dialing the various signaling pathways, dialing a developmental pathway that increases yield and photosynthetic capacity. At the same time, you've got the flexibility to dial up and dial down the drought resilience or the water locking resistance with the supergenes. So it's that those machineries are all in the plant. What's not there yet is our ability to individually dial the different control points in the plant. And that's what's coming. The knowledge is close for a lot of us. How well we stack it together, it will be the challenge for a lot of us. We need to solve though, otherwise it's going to be bad.
Yeah, so it was really, really great discussion. So I wish I could continue this discussion, but every good things come to an end. So I will now hand over to Mary Williams.
Thank you, everyone. Thank you, especially Barry and Andrew, for staying up very late. I believe it's tomorrow for you now. So, but it was an exciting talk. Honestly, it makes me kind of wish I could start over as a PhD student and have another, 30 years ahead of me to play with the rheostats, because it's really clearly exciting work. Thank you, Damaris. Thank you, Jesse. Thank you, Katub. Nan, would you like to say anything?
No, just thanks to all of our presenters and to Kutub for moderating, and to all of our participants for viewing. Yeah.
And we encourage you to read the papers.
Absolutely.
Thank you, everyone. Bye-bye.
Bye.
Bye-bye, everyone. Bye.